Hello, my friends, and welcome to our video lecture on topic C4.1, Populations and Communities. Our guiding questions for today: how do interactions between organisms regulate sizes of populations in a community, and what interactions within a community make its populations interdependent? Today's objectives: we're going to look at some vocabulary, talk about a couple techniques for estimating population sizes. We're going to define carrying capacity. We're going to look at some density independent and dependent factors that affect population sizes. And then we're going to look at some intraspecific and interspecific relationships. We are going to define top-down versus bottom-up population controls, define endemic, alien, and invasive species, talk about what even is allelopathic and antibiotic influence on population sizes. We're going to wrap up with some chi-square statistical analysis of interspecific competition. Little bits of vocabulary to get started. Species are groups of organisms that can interbreed to produce fertile offspring. Populations are groups of organisms of the same species living in the same area at the same time. Communities are groups of populations, different species, living in the same area at the same time. Again, populations are groups of organisms of the same species living in the same place at the same time. Let's talk about how we could estimate the size of this crab population. This little guy I encountered when I was doing my Fulbright seminar in Thailand in the summer of 2024. He was on the beach in Hua Hin, which is just a little bit south of Bangkok. This guy was maybe 1 centimeter tall at the most, clearly put on a big defense though, despite his small size. These crabs live in these tiny little burrows along the beach. And as I walked along the beach, they would scurry into their burrows to hide from my giant feet. And if we wanted to estimate the population of these little crabs on the beach in Hua Hin, we have a couple options. We could do some systematic sampling. Systematic sampling would be, maybe I'm going to lay a tape measure along the beach and I'm going to count all the burrows in a square decimeter area every 5 meters along the beach. So this is very systematic. I have laid out my plan. It's very organized. Here we go. One of the drawbacks to this systematic sampling is that we might, drag our meter stick along a place that we wanted to count. Maybe that's where there were more burrows, so we decided to put our line there. Another way, perhaps a little bit better, is some random sampling. And random sampling is going to allow us to pick random spots in the beach, and then we would count the burrows. And then we're going to use those samples to infer the total population size. Why not just count all the crabs on the beach? Because that would take a really, really long time. It would be very difficult, if not impossible. Why do we care about population sizes? They give us a really good indicator of the health of ecosystems. So let's look at a couple ways that we could randomly sample some population sizes. The first technique that we are going to look at is quadrant sampling. In quadrant sampling, we are going to take the total area that we want to sample from. In this example, we have a 10 meter by 10 meter space. We're going to divide that space up into increments that make sense. In this case, 1 meter increments make sense. We are then going to use a random number generator. It might be an app that we find online or some other method to randomly choose the coordinates where we're going to count organisms. So let's say that our random generator gives us a coordinate of 5, 1. We will go over 1, 2, 3, 4, 5 along the x-axis, up 1 on our y-axis. Then we will lay down our quadrant. Quadrant is this stiff frame that has controlled dimensions, and then we will count the organisms of the species that we're looking at that are just in that quadrant. And then we'll use our random number generator to pick other spots, and then we can use this to make some inferences. We'll build a ratio, a proportion up to decide the total number of organisms in the whole space. The benefit of using this random number generator is that the researchers aren't going to be picking where they want to count. Instead, we should get a pretty good representative number of the organisms in this population. that quadrant sampling technique is really good for sessile organisms, organisms that do not move. If, however, we have mobile organisms, organisms that can move, quadrant sampling is not going to work very well because we might have the same organism get counted more than once if it's moving from one quadrant into the next as we're counting. So we have this other technique for our mobile organisms. It is capture, mark, release, recapture. So let's say that in this example, we are going to capture this first group. We have 1, 2, 3, 4, 5, 6, 7 mice that we capture the first time. We're going to mark them. Notice the little yellow tags on their heads. Sometimes we paint a color on the organism. Sometimes we put radio trackers on the organisms. We're going to somehow mark or tag these guys. We are then going to release them back into the wild. A little bit later, we're going to do a second capture, our recapture. In this example, we have recaptured 1, 2, 3, 4, 5, 6, 7 organisms. So 7 guys from our recapture. Notice that 2 of the guys in our recapture group were tagged in that first capture. We are then going to use these numbers in this equation, the Lincoln index equation, to estimate the population size. We had 7 mice in our first capture. We also had 7 mice in our second capture. 2 of them had been marked in the first capture and then recaptured. So 7 times 7 is 49, divided by 2 is 24.5. We can estimate 24 or 25 total organisms in this population. One of the limitations of this technique, the capture, mark, release, recapture, is that the tagging, the marking can cause injury to the organisms, which might make them more susceptible to predators. If it is some kind of color that is the mark, then it might make them more visible to predators, which is going to, of course, lead to skewed numbers. This also is assuming that there's no immigration or emigration in this population, which most of the time we're going to have lots of immigration and emigration, especially in our animal species. As we estimate population sizes, we can graph them along time. And we have found that in many ecosystems, there is a carrying capacity. Sometimes we denote that with the letter K. That is the maximum population that is stable, sustainable in an environment with the resources that are given. So this place of stability where we don't have a lot of population growth, it's not going to be exactly the same number. There will be some fluctuations up and down, but the average number is going to stay about the same. Some of the things that are going to lead to what that carrying capacity number is for plants is availability of water, availability of light, and the availability of nitrogen in the soil. Any one of these will cause that carrying capacity to be a little bit higher or a little bit lower. For animals, water is again one of these limiting factors, one of the things that is going to determine what carrying capacity is equal to, the space that there is for breeding, how much food there is, and then, especially if we're talking about aquatic animals, the dissolved oxygen content of the water that they're living in. We often describe these growth curves that get us from initial population numbers to the carrying capacity. We call it sigmoidal because it looks a little bit like an S. And here we're going to have this initial slow growth phase, which has some exponential growth. We're going to go from exponential phase to a transitional phase where growth is slowing down. And then we're going to be in our plateau phase where we've reached that stable number, which is equal to carrying capacity, or K. And this is our 3 phases of sigmoidal population growth. There are many factors that influence what population sizes can be. Some are density independent. This means it doesn't matter how many organisms are in the population. Floods and forest fires, other natural disasters, or human changes to an environment are going to be density independent factors that affect population sizes. We're going to talk more about some density dependent factors because these are relationships amongst groups of organisms in an ecosystem. And they are often negative feedback factors. Competition is going to lead to some negative feedback. The higher the population number, the more competition there is, that increase in competition is going to bring those numbers back down. As there are more organisms available to be hunted, more are going to be hunted. That's going to bring those numbers back down. If we have a high density population, we're going to have more spreading of disease, more spreading of parasites and other pests. Higher density of the population is going to increase these factors, which will decrease the population. And these are going to bring us to that carrying capacity. There are other factors that regulate population size that are density dependent that can be positive feedback. Of course, the more organisms there are, the more organisms are available for mating, the more babies will be born. But generally that's going to lead to a lot of these negative feedback loops that will bring those population numbers back down. Interactions amongst organisms that regulate population sizes can be intraspecific. Intra means within, so we're talking about organisms in the same species. Here we have some birds competing for food. These guys are all the same species of bird. We can also have interspecific interactions. Inter means between. So now we're looking at between different species, and here we have some different species of birds competing for food on the bird feeders. When we're talking about intraspecific interactions, we are looking still at populations, these groups of organisms of the same species living in the same place at the same time. When we're looking at our interspecific interactions, now we're talking about communities. Communities are our groups of populations, different species living in the same place at the same time. Other examples, we could have flowers of the same species competing for pollinators. That would be intraspecific. We could have flowers of different species competing for pollinators. That's going to be interspecific. Some of our interactions that regulate population sizes can actually be cooperative, leading to increased population numbers. Here we have an intraspecific cooperative behavior. These meerkats are huddling together to keep each other warm. That's going to increase their fitness, increase their survivability, and therefore increase their population size. Here we have an interspecific cooperative relationship where these butterflies and these flowers are helping each other survive. The butterflies are getting nectar. There's lots of sugar, lots of energy, ATP production available for those butterflies with that food. And then the flowers are getting pollinated. Their sperm cells in the pollen grains are getting spread, which is going to increase genetic variation in their species. So both species in this community are going to be benefiting. Therefore, it's an interspecific cooperative behavior. Because we humans do love to categorize things, we have some major categories of interspecific relationships. Inter, remember, is between, so we're talking about relationships between different species. So we're talking about community interactions, not simply population interactions. One example of an interspecific relationship is herbivory, where we've got herbivores eating plants, so they're feeding on the producers. Predation, here a predator is going to kill and eat its prey. Resource competition, different species are using the same resource, whether that's the same water or the same food or the same environment, the same niche in an environment. We could have mutualism. Here, two species are both benefiting from a relationship. Parasitism, similar to predation, except there's no outright killing of the prey. Instead, it's a slow feeding off the host. The host might eventually die, but it's not an outright immediate killing and eating. Pathogenicity is similar to parasitism, except here we have a pathogen, a disease-causing agent living in the host, causing that disease, getting resources from the host. Consider pausing the video and seeing if you can identify the 6 different categories of interspecific relationships that are illustrated in these images. Here we have resource competition. These trees are competing for sunlight, and so some of them will try to grow much taller to get more sun. these guys here are being shaded, they're not going to get as much light. They won't be able to do as much photosynthesis. They're not going to be as healthy because of this competition for sunlight. Here we have some pathogenicity. This is potato blight, which is a fungal infection on these potatoes, which is going to lead to the fungus gaining resources and the potatoes not doing so well. Here we have some mutualism. The bee is getting nectar from the flower. The flower is going to be pollinated. The pollen grains have sperm cells in them. Those sperm cells can then go fertilize other flowers, which is going to help the reproduction of these plants. Here we have some herbivory, of course, these deer eating leaves. And then this is maybe one of my favorite kinds of predators. This is the fierce ladybug. If you are not in the United States, you might call it a ladybird instead. And it is a carnivore. It is eating this critter called an aphid. This image is also showing us parasitism. Aphids are the mosquitoes of the plant world. So what these aphids do is they stick their stylets, their little noses, into the phloem of the plant and suck the sap, the organic materials, out of the plant, just like a mosquito would suck human blood. So here we have a parasite parasitizing the plant, and then here we have predator eating its prey. We're going to look at a few more examples of interspecific relationships, these relationships between different species of organisms, in a little bit more detail. We're going to look at 3 examples of mutualism because it confers such an amazing evolutionary advantage to the organisms involved. Then we're going to look at a way that predator and prey numbers interact with each other. So first up, this example of mutualism, we have here a leguminous plant. Legumes are beans and peas. And what's pretty cool about these guys is they frequently have these nodes on their roots. These nodes are filled with bacteria. These bacteria specifically are called nitrogen-fixing bacteria. Nitrogen in the air is in the form of N2 gas molecules. There is a triple bond between the 2 nitrogen atoms in a nitrogen gas molecule. And so even though 78% of our Earth's atmosphere is composed of nitrogen gas, it is entirely unavailable to plants. So these bacteria will fix the nitrogen. They can actually break that N2 bond and then convert the nitrogen gas into either ammonium or ammonia. That ammonium is available to the plants. The plants can use that ammonium to make nitrogen-containing compounds like DNA and protein. Some nitrogen-fixing bacteria will make ammonia instead. That can then be nitrified into nitrates, which can also be taken up into the plants. There are other bacteria that will take those nitrates, turn them back into, eventually, nitrogen gas, and so this is our nitrogen cycle. The plants' benefit in this mutualistic relationship is that they get the nitrogen that they need. It prevents them from being nitrogen deficient. The benefit to the bacteria, they don't like oxygen, and so getting to live in these little nodes on the plant roots, they can be away from the oxygen gas. They get protection from the plant. The plant provides them with some glucose so that they can live and make bacteria babies. Our second example of mutualism is between orchid plants and these fungi called Mycorrhizae. So Mycorrhizae can live in the roots of orchids. Orchids frequently will not put their roots into soil. They will not get nutrients from the soil. Instead, the mycorrhizae will get nutrients from decomposing detritus around the plant or by parasitizing other plants that are near the orchid. And then the mycorrhizae is going to provide nitrogen and phosphorus and water to the orchid. The orchid is going to give the mycorrhizae a place to live and then send them some sugar if they're photosynthesizing. Some orchids don't even photosynthesize because the mycorrhizae are going to provide all the nutrients that they need. Another example of this same relationship is in orchid seeds. When we talked about plant seeds, we talked about the endosperm, the cotyledon that provided nutrients for the growing plant embryo. Orchid seeds don't have that. There's no endosperm. There are no stored nutrients in the seeds of orchids. Instead, the hyphae of these mycorrhizae are going to grow into the seed, And then the mycorrhizae, the fungus, is going to provide the seed with everything that it needs, the water, the nutrients, that will allow the seed, the embryo in the seed, to develop. And our final example of mutualism in a little bit more detail is the relationship between zooxanthellae and coral. Zooxanthellae are these microscopic photosynthetic plankton that will actually live inside the coral. The coral is going to provide carbon dioxide from its own cellular respiration. That's going to provide a place to live. The coral will grow fairly close to the surface of the ocean so that the zooxanthellae have lots of access to sunlight. In return, the zooxanthellae provide sugar that is produced during photosynthesis, other organic compounds, and oxygen gas that is released as the waste product of photolysis. One way that we know that corals are not doing well is when they are bleached. And coral bleaching is actually the dying or the expulsion of the zooxanthellae. The zooxanthellae, guys, these are the ones that actually provide the color to the coral. And so the bleaching or the loss of the zooxanthellae is an indicator that the coral is not healthy or could in fact be entirely dead. What can cause this? Oceans becoming a little bit too acidic. Their pHs go down, become more acidic. This can cause coral bleaching. Another cause is rising temperatures in the ocean. And as promised, we're going to look just a little bit at the influences that predator and prey numbers have on each other. This is an example of those density-dependent factors that influence or regulate population sizes. There are normal seasonal fluctuations in predator and prey numbers. So perhaps this prey has a springtime breeding season, and so in the spring we're going to have more and more prey animals being born, which is going to increase their numbers. However, that is going to increase the supply of food to the predators, and so they're going to be more fit, they're going to have more babies, but then more prey are getting eaten, and that's going to cause a decline in their numbers, which means that there are fewer resources available for the predator. Their numbers will also decline. This seasonal fluctuation is totally normal, and these fluctuations are right around the carrying capacity, or that number K, that is indicative of a stable population. Sometimes, though, things happen that lead to some pretty unstable number changes. This graph is showing us the outbreak of a disease, scabies, in foxes in Sweden in the 1980s. So scabies led to the death, the unfitness of lots of foxes. And so the fox numbers declined quite a bit. Because the predator numbers were down, there wasn't this regulation of the hare numbers. And so the rabbits were able to make so many babies because the foxes weren't around to eat so many of them. This led to, interestingly, some intraspecific competition. So there's more competition within the mountain hare populations, since the red fox predators were not regulating what those population numbers were. All of these community interactions, these interspecific relationships we've been talking about, are going to regulate population numbers. We can call these controls of population numbers either top-down or bottom-up. In top-down, literally the top of this energy pyramid, the top carnivores are going to regulate the numbers of consumers, which is going to regulate the numbers of producers, which is going to regulate the numbers of decomposers that are in the environment. In bottom-up control, the soil nutrient availability, which depends a lot on those decomposers, is going to have an effect on how much producers can be in the environment, which is going to affect the number of herbivores, which will affect the number of consumers. Just a little note on this pyramid. I do kind of like how it included the soils decomposers so that we could talk about from literally the bottom of this pyramid. But just know that in IB world, we don't like a triangular pyramid. We prefer a block. And so if and when you are asked by IB to draw a pyramid of energy, draw it in stacked blocks, not as an actual triangle. Good. All right. For our community interactions, for our interspecific relationships to regulate population sizes, we need endemic species in the populations in the communities. Endemic means native. For example, this koala is native or endemic to Australia. It only lives there. Any other place we find it should hopefully only be in a zoo. However, sometimes species are introduced to new areas, and at that point we're going to call them alien species. Sometimes those alien species can become invasive if there are no density-dependent factors regulating their population growth. An example of an invasive species are these zebra mussels. They are living almost everywhere now, and they are overwhelming the marine habitats because they don't have predators away from their endemic environments. So they become quite invasive. There aren't any natural predators. There aren't any density-dependent factors that are regulating their population growth, and so they're just everywhere. And humans are working really hard to try to control their numbers, but that's very difficult to do. Another example of an invasive species, one that was alien and then didn't have predators and is now invasive, is this red lionfish. It is endemic or native to the Indo-Pacific regions of the world. In that part of the world, there are predators that control their numbers. However, there was a hurricane that caused some damage to an aquarium in Florida, and a bunch of these guys escaped. And so now there is a large red lionfish population in the waters around Florida because they don't have any natural predators there. It's now an invasive species. There are no density-dependent factors regulating their population growth. And two more little examples of community interactions that regulate population sizes. The first one we're going to look at is the production of antibiotics by fungus. So this is a penicillium fungus that is releasing penicillin, the chemical that inhibits bacterial growth. This is preventing the bacteria from competing with the penicillium fungus for resources. Of course, as humans come and steal these antibiotics from the fungus, we use them to treat bacterial infections in us, which is nice until we have all kinds of antibiotic resistance evolving in these bacteria populations. Another example of regulating population growth is allelopathy. In allelopathy, what we have is some plant that secretes a chemical into the soil that is going to inhibit the growth of other organisms that come into contact with the chemical in that soil, so that the tree is not competing for resources with plants nearby. Allelopathy, antibiotics, chemicals released by living things to inhibit the growth of other living things. And since we started our video lecture with some statistics, that inferential statistics that we do in quadrant sampling and in capture, mark, release, recapture, it's only fitting that we would wrap up also with some more statistics. Here we're going to look at how we can use chi-square to investigate interspecific relationships. We can count organisms either in the field, out in real nature, or in the laboratory. Both have advantages and disadvantages. And we're going to put together some hypotheses. We're always going to have a null hypothesis, or our H naught. And this is going to be that there is no relationship, that we don't see any influences from one species on the other. Our alt hypothesis, and we can actually have more than one. We could have hypothesis 1, 2, 3. This is where we're looking at the actual presence of some kind of relationships between these different species. How are we going to do this math? Number one, we are going to count observed frequencies of the two species, probably using quadrant sampling since we're looking at trees here. And what I'm going to do is figure out in how many quadrants in my environment do we have both trees present. And we counted 25 quadrants have both species of trees. We're going to figure out how many quadrants in our environment do not have noble, but do have the grand fir trees. We'll do the same thing. How many do have the noble, but not have grand? We're going to figure out how many quadrants have neither of these species. Now we're going to do some math that we'll then use to calculate our expected frequencies. First step, we're going to do some addition. So we're going to do 25 plus 30, that's going to give us 55. 45 plus 50 is going to give us 95. We're also going to add across. 25 plus 45 is going to equal that 70. 30 plus 50 is going to equal that 80. And if we did our math correctly, these guys should add up to the same number as these guys. And they do. Hooray. So how are we going to use these values to calculate our expected frequencies? Step number 1, to figure out our present, we're going to use the total columns for present and present. Let me erase these scribbles so that we can actually see those numbers. So we're going to go across to the present total, 70. We're going to go down to the present total, 55. We're going to multiply those two numbers and then divide by the grand total of 150. We're going to do the same thing. We're going to go to absent total 95, present total 70. We're going to multiply 70 and 95 divided by the grand total to get this number. This number. We're going to continue doing the same process. So we'll do across absent is 80, down present 55, multiply those, divide by the grand total, that gives us this number. We're going to do the same thing for that final little piece. We're going to go absent, down, absent, across, multiply those, divide by the grand total, and then we're going to again add. So 29.3 plus 50.7 should give us the same number that we got up there. Same thing here, and that's a good check for our math. So now what we're going to do is regular old chi-square. So we're going to have our two conditions. We're going to do observed, expected. We're going to calculate chi-square, observed minus expected squared divided by the expected. We will do that for present and present and absent all over the grid. And then we're going to add up all of those numbers, and that's going to be our chi-square value. We will compare that to our distribution table to figure out what even is our p-value. And the p-value for 0.056, chi-square is somewhere in here. 0.056 is between 0.10 and 0.01. So our p-value is going to be something between 0.9 and 0.75. That's too big. That is going to be a failure to reject the null hypothesis, which means that there isn't actually any relationship amongst these tree species. They don't care where each other live. Good? We're going to do this in class. So if you're like, what just happened? No worries. We're going to practice. But my friends, get excited because we have arrived at the end of our video lecture. We talked about our vocabulary, those super important 3 words: species, population, community. We talked about how we can estimate population sizes with quadrant sampling or capture, mark, release, recapture. We talked about carrying capacity. We use the letter K to talk about that stable number that a population can grow to and still be sustainable in its environment. We talked about our density independent and dependent factors that regulate population sizes. We talked about intraspecific relationships within a single species, interspecific relationships between different species. We talked about that top-down, bottom-up population control. We talked about species that can be endemic. This is good because they're native and they belong. We talked about alien and invasive. Invasive species are really bad. We talked about the allelopathic antibiotic influences on population sizes, and then we talked about chi-square, a lot of math, and how we can use that to determine if there is interspecific competition occurring or not. Great work today. Keep it up.